It is, according to Kenpia, the prevalence rate of HIV at Nyeri County was at 5.1% compared to the national prevalence of 4.10%. So this was quite a big problem, and so we sought to establish the factors that determine the patient's adherence to the antiretroviral therapy, their clinic attendance, and the rate of defaulting or their loss to follow-up. Also, a notable proportion of the patients attending the clinic there who had a high viral load were noted to be chronic defaulters to the clinic, and so it was quite a big problem to manage the condition. Also, this led to poor patient outcomes such as mortality and morbidity, which I will expound on later. Also, in the justification of our study, as I had stated earlier, the pandemic continues to be a global burden, leading to significant mortality and morbidity. So adherence to the medication helps in viral suppression and improving the quality of life of the affected individuals. As healthcare workers, we advocate for 95% adherence to the medication. Also, non-adherence brings about clinical failure, high viral load, and consequently increased mortality and morbidity. And the studies on adherence and retention of the patients at the clinic there, they have been under-researched. When it comes to the objectives of our study, our broad objective was to assess the determinants of adherence to ARTs and patient retention. Specific objectives, we sought to compare adherence and retention according to the social demographics. Also, we sought to establish the determinants of adherence to the ARTs, rate of retention, and over a period of 1 year among people living with HIV and AIDS. And also, we sought to establish the relationship between non-adherence and loss to follow-up and poor patient outcomes. This is our conceptual framework as indicated in the slide over there. So in our independent variables, we had the social demographics, the social support, and the financial support. The financial support as our independent variables. Our dependent variables is the adherence and the retention. So in our methodology, it was a descriptive cross-sectional study design, which was carried out at the comprehensive care clinic at the hospital. And our study population was the patients who attended the clinic and they're aged 18 years and above. And we used the simple random sampling method to recruit participants to our study. In our data collection methods, we used structured questionnaires which we administered to the patients. Also, we reviewed the existing databases at the clinic, the hard copy and the soft copy. And also, we used the more risky medication adherence scale tool. And to calculate adherence of the participants to medication, we inquired from them whether in the past 7 days they have missed a dose of their medication. So to calculate the adherence, we take the 7 days, we minus the number of missed doses over 7 times 100 to get the percentage adherence. We Imara kama Simba. Thank you.
Thank you. Wamesimama kidete. Thank you so much for that presentation. That presentation was very important. Contracting HIV/AIDS is not the end of life. We need to ensure that these patients adhere to medication, that they don't face complications, and we do not lose them to the disease. Jesse and the team, thank you so much. Time is not really on our side. We are getting time bad as minutes go by. So I think I will want to give an opportunity to our sponsors, some of our sponsors who came through. This event could not have been a success without them, and it won't be good if I don't acknowledge their presence and acknowledge their support. So at this juncture, I think I'll invite someone from Afia Record to just give us an address and tell us about the organization. Thank you.
Hi everyone. Are you happy to be here? Are you learning the way I'm learning? Yes. Okay. I'm from an organization called Afia Record. So Afia Record is a patient-driven health data platform. So we connect all the various ecosystem to patient via our platform built on deep technology, which is blockchain and artificial intelligence. So we build tools that place the patient at the core of healthcare delivery. Our mission is to give patients ownership of their health data and support various healthcare providers to support with the right tools to digitize and generate revenue and also connect with the patient. So for Afia Record, we believe that treatment happens in the hospital, and we believe that treatment happens in the hospital and treatment happens outside the hospital. We have two core products. We have B2C and we have also B2B. B2C is a standalone patient portal for the patient to use to store their health information and share with their doctors. And also we have the B2B, which is to digitization for hospitals when they have records and it's analog, how you can be able to digitize such platforms, and also for pharmacies. Yes, so I will invite everyone to go to myrecord.com to check it out and give us a feedback. So I want to agree with Dr. Murugu, something that he said about research. So I will urge everyone from the first years to the last year of medicine to participate in research. Because when you participate in the research, you become a critical thinker. What does that do when you become a critical thinker? You bring solution. You bring innovation, something that did not exist in the first place. So you become an entrepreneur. Ahead of being a doctor, you're also an entrepreneur. That's very exciting. So I will urge everyone to be part of research. It's amazing. I would like to congratulate Jkuat for this initiative. Congratulations. We are excited as Afia Record to collaborate with the research institutions that improves quality healthcare and ensures quality of life of all the patients. Thank you so much and congratulations once again.
Thank you so much. I hope the relationship between Jkuat and Afia Record will maintain from now on. Jkuat official mesikia veni watetea. So thank you. I think I'll also call the other sponsor, Exela. Okay. Victor wants to give gifts. Victor is feeling good. Victor is feeling nice. Okay, Hannah, please. I'm taking my coffee. Don't worry. Lunch is here. Lunch. Actually, we're preparing lunch for you people. So just clap if you're told to clap, kindly. Yeah, so I'll give an opportunity to Excella Biotechnologies Limited. I think they have a representative around for their address. Thank you. Yeah, there's a presentation. As we wait for that presentation to be put up, how many are learning? Wanja, what have you learned today? I'm putting you on the spot.
Thank you. I've learned that microbiologists in the lab are the source of AMR. But seriously, all of us are the cause of AMR, all of us, one way or another, and in any position that you can get a chance to bring change, may you be the change. Yes.
Thank you. Let me give a chance to somebody in the back bench. Nimona some 2-3 people wakidoze off. Let me see. Who wants to say something from the back bench? What have you learned? My brother, Gitonga.
Thank you very much. So I've learned about the post-abortal care, where you find most of the patient, despite receiving that, let's say they have come in with an incomplete abortion and they need that manual vacuum aspiration. We as doctors and as also other healthcare stakeholders, we just act basing on the let's say the medical part of it and not the psychological part, which is very important because our key role is not just to receive that patient back and back again. We just have to deal with the patient wholesomely. Yeah.
Why should we lose a patient from the OBS gyne for them to be admitted into a psychiatry ward? Doesn't make sense, right? Yeah, so let's not neglect psychological support. Hapa katikati, Owen, do you feel like going? Feel like saying something? Acha, unaniangusha. You're making me look bad. Jonah? Akianan, you're making me, there are MKU students and KU students here. Anybody from MKU? Guys, what have you learned, my brother?
Okay, thank you. I've learned that we're really integral in making a lot of innovative but simple changes. Also, the fact that we are supposed to just start, get somewhere, look for help. There's always help. There are many platforms to reach out, and also that role of being a steward. I think it's something that I didn't know, that there was a diagnostic stewardship and infection prevention stewardship, which is really interesting.
I salute each and every person who has given me a response. Student, after this, lunch. We're in a pair. So many students. Irene, take it away. Thank you.
Good afternoon. I'm Irene Muriuki from Excella Biotechnologies. Allow me take you through what we do and who we are. So Excella Biotechnologies Limited is a reliable solutions provider focusing on distribution of leading brands of life science products and molecular diagnostic by research and diagnostic laboratories. So we have various partners. We have Altona Diagnostics, Pcr Kits, Beckman Coulter, and ESCO Life Sciences. So our mission is to build long-term relationships with our customer and clients and provide exceptional customer services by pursuing business through the latest innovation and advanced technologies. So allow me tell you more about Altona Diagnostic, one of our brands. So products we offer from Altona Diagnostics, we have RealStar real-time PCR kits. These are molecular PCR kits. So RealStar real-time PCR kits are designed for quantification, qualitative detection, and or differentiation of pathogen-specific RNA or DNA. So we have a picture there. So our kits are designed as such, and they are packaged in vials, like you can see, and they are coded for ease of use. We have different types of real-time PCR kits for the detection of tropical and other viruses, bacteria, and parasites. So all these are the kits that we have available under tropical and other viruses, bacteria, and parasites. And under this category, we've been supporting the East African communities for their mobile labs and also the border surveillance, and we're also supporting them for the Ebola outbreak that is happening. So for respiratory viruses, bacteria, and fungi, we have different PCR kits as well. We have the SARS-CoV-2 RT-PCR kit, which is the COVID testing kit. We have others, as you can see. I won't go through all of them. We have a category for human adenovirus, human herpes viruses, and human polymerviruses. For that, we can also see we have the CMV, we have the human herpes viruses, and the rest. We also have a category for enteric viruses and bacteria. Also, we can see the list. We also have a category for blood-borne viruses. We also have a few. So allow me move to the next part now. We have Beckman Coulter Life Sciences. Products we offer from Beckman Coulter Life Sciences. We have the biotech business unit where we offer centrifuges. You can see the picture there. Those are our centrifuges, but we have other varieties as well. We have automated liquid handlers for your sample extractions and all that. We have particle and cell analyzers. We have genomic reagents for your extraction, purification. Flow cytometry business unit, we have flow reagents, analyzers, sorters, and softwares. I'll focus kidogo on flow cytometry solutions. So we have a few solutions for flow cytometry. We have the Aqueous SL, which is an automated load and go flow cytometer, combining sample preparation and analysis in one platform. So applications are in identification and enumeration of T and B and NK lymphocytes. It has one laser and 4 colors. So I think, Dr. Ri, I have a solution for you for HIV research. We have a Cytoflex, which is a research flow cytometer. It is superior, sensitive, and resolution throughout all configuration. Give it the edge over other cytometer systems, 4 times its size. So as you can see from the picture, it is very small, but the idea is big. So application in CD4, CD8, leukemia and lymphomas, stem cell research, among other applications, with up to 6 lasers and 21 colors. That means you can do research for up to 21 parameters. We have the Navios EX, which is a clinical flow cytometry used for CD4, CD8, leukemia, lymphoma, stem cell research, among other applications. It has 3 lasers and 10 colors. These we have in the country, one at the Aga Khan University Hospital. So for the cancer research or clinical, you can use the Navios. We also have the DX Flex, is a clinical flow cytometry used for CD4, CD8, leukemia, lymphoma, stem cell, among other applications. It has 3 lasers and 13 colors. We also have single antibodies for the flow reagents. So in case you need to do your own panels, you can combine as you wish. We have the Kaluza-C analysis software, which is an advanced software for analysis. So it helps our pathologists. We have the ClearLab 10C system, which is a flow reagent. So these are pre-mixed antibody panels, so you don't have to mix manually, and that reduces the potential of human error. Also, it helps with pipette. It also helps you with you have tubes that have your mixed antibodies, so you can have tubes for the B cell, T cell, myeloid 1 and 2. Also, the kits comes with compensation kits and setup kits. It also have controls and also the Kaluza system. The other brand is Biocatis. So with Biocatis, we have the IDELA platform. Revolutionary, fully automated real-time PCR based on molecular testing system designed to offer results in minimum amount of time. So this is a very interesting platform because as you can see, I'm not sure if it's so clear, but it is a fast PCR solution. You don't have to do the extractions. You don't have to do the master mixing. You just need a cartridge. So you extract your sample, you load it to the cartridge, and you get your results. So in under 2 hours, you have your results. So these are is a molecular solution that we have. So what does IDELA mean for cancer patients? So we have various rapid treatment initiations, and we have different molecular diagnostics. We have myeloma, colorectal cancer, lung cancer, and different other mutations that you can do with the IDELA. So for the oncology test, you also have various tests that you can do with the IDELA. And because of time, I'll finish there. In case you have any other questions or you need clarification, you can meet me outside there and I'll explain further. Thank you.
Thank you so much, Irene from Excella. I think I'll call the beautiful Sarah to gift Irene from Excella. Photographer kwa wapi? Where is the photographer? Photographer wenda wapi bana? Bro. Yeah, thank you so much for that presentation. I think it's about that time. Lunchtime, say. So, it's lunchtime. So, I'll call Victor to give us the directives on lunch. But thank you so much, everybody. Thank you so much to the photographer. Thank you so much to our tech savvy, Mr. Pius Murangiri. Mr. Pius Murangiri is my friend, is my classmate, also a research enthusiast. Thank you so much. Yeah.
Asante sana Eldon. So I know it's the time when you guys are hungry, so I'm going to give the directions. Our guests, you're going to go to the VIP lounge. We're going to serve you there. Then, CC delegates, I want us to go through this door, proceed straight, take your left, then wash your hands, pick up plate, then serve, proceed inside again. Tumeelewa? Walk out through this door, left, wash your hands, pick up plate, serve. Sindio? Asanteni sana.
Why do you look at me this way, oh daddy? Oh daddy. What is it that you wanna say, oh shitty? Oh shitty. Yeah, God put you my way.